So I'm very, very happy to have Qian with us today because she's really an absolutely amazing bioinformatician. And now she's an assistant professor at University of Texas Southwestern, and she continues to work on protein-protein interaction screen, silico, and she will tell us more about what she's currently doing. Qian, I'm going to stop sharing, and now you can share your slides.
Anastasia, maybe you can tell people how they can interact, with the questions.
Yes.
And so on.
Yes, so there are two ways to interact. So you can post questions in the chat. And since we are a smaller group today, we can also, at the end, you can raise your hands and we're going to invite you to ask a question. So Qian, I'm looking forward to your talk.
Thanks, Anastasia, for the wonderful introduction. So sorry, I did mess up the time a little bit. I think I probably marked on my calendar before the time changed. And although Anastasia reminded me it's 10, then I still have 11 marked there. So I thought, oh, it's 11. Very sorry about that. Hope I can do a better presentation than I usually do to make up for my mistake. Well, so I'm going to talk about the in silico screen of protein-protein interaction. This is the direction I started in the Baker lab and continued it as I came to establish my own lab. So basically, the basic principle of this is probably already kind of well known among people, where it is basically from the co-evolution in sequence we can predict the 3D structure. And so basically, if you look at the protein sequence alignment, you may see some positions they change in a correlated way, and we call this phenomena co-evolution. This usually arises from some interaction in the 3D space, which will constrain the evolution of the two residues. And that's why using such information, we can deduce which residue should come close to each other in the 3D space. But the method to detect co-evolution has been evolving over the last 20 years. So in about 20 to 10 years or so, co-evolution was usually analyzed in a very simple way using local statistics that look at one pair of residues at a time. So basically you want to see if just two positions, if they have some dependency upon each other. So that method, it did reveal some insects, but it's not good enough to look at, to predict 3D structure, because if you just look at one pair of residue at the time, it's hard to distinguish which pair of residue are directly interacting or which pair they are showing some co-evolution just due to some indirect transitivity of this co-evolution signal. So, and then as time evolved, about 5 to 10 years ago, people start to look at this data from a more global view, where they look at all pair of residues from one protein together. So this global view allows them to deduce which pair of residues are directly interacting, and they should form a contact in the 3D space. So this did lead to a small breakthrough in the structure prediction field, where people can predict the complicated topology of a protein accurately without using of known protein structures. So, and then just about 2 to 5 years ago, so there is yet another breakthrough in this field when people start to introduce deep learning method to look at this data. So compared to the global statistics that can look at one protein at a time, deep learning method actually can look at all proteins. So all proteins with known structure, it learned the rule from all those proteins. That's why it can summarize the rules and help you to deduce the signal from your target protein better. So this did introduce another breakthrough, small breakthrough in the accuracy of the method. But the final big breakthrough is really becoming like it's very well known even beyond the field. This is introduced mostly by AlphaFold from DeepMind, where they use the most innovative and specially designed deep learning architecture to this problem. And this method apparently in somewhat a practical level solved the single domain protein folding problem. And So one important fact for me is as the method improve, the number of sequences you need to make an accurate prediction is decreasing drastically. So in the beginning, while in the mutual information time, even if you have a lot of sequences, you are not going to make a very accurate prediction. But starting from the global statistics, you can make very accurate prediction if you have a lot of sequences. So this is like I say 1,000 to 10,000. This is after reducing the redundancy. So just diverse sequence, if you have like on that order, you can do a good prediction. But up to now, if you have like 100 diverse sequence, then you can already make a very accurate prediction. So apparently this latest breakthrough is led by AlphaFold, made by the DeepMind team and my postdoc advisor, so who used to be a leader in the field and who will never give up. Also quickly, together with us, made our tool inspired by AlphaFold, but using somewhat different architecture, which we call Rosetta Fold. And my take of this, these breakthroughs are basically, I want to make it like in parallel with this sequence genomics breakthrough. So sequencing genomics was also launched like 10 to 20 years ago, where people start to sequence the human genome. And of course, only now they claim that the human genome is complete. But in the course of sequencing human genome, they invented this next generation sequencing technique, which drastically decreased the cost to get any genomes. So as a grad student, I actually mostly work on genomics, although I'm from a very computational protein structure lab, but my main contribution to the lab was to start a genomic direction in the lab from scratch. So my grad school research greatly benefited from next generation sequencing as many other people. So, and but this structure prediction revolution, my view is okay. So before there is this structural genomics, and by itself it produced a lot of data, but it didn't yield a technology breakthrough that can drastically reduce the cost of obtaining 3D structure. But together with artificial intelligence, that time came, and now everybody can easily get the 3D structure on a genome scale. So then this obviously will lead to some change, like AI-driven drug discoveries and the studies of diseases and evolution and many more scientific questions. So my own research isn't so much focused on predicting the structure of individual proteins. I'm more interested in predict protein-protein interactions. So this is just a slight generalization of the single protein structure prediction problem. So the task now, instead of studying one protein, you are studying one pair of protein. And the task is just to see that whether the evolution of protein A would affect the evolution of protein B. Then there's a small challenge that is to pair proteins that are supposed to evolve and function together from families A and B. Then you want to create a paired multiple sequence alignment to convert this problem, this two protein problem, to like a one alignment problem. So, and how do we generate these paired alignment? So we made a very simple assumption that the proteins of the same species, they should evolve and function together. So let's say, so I'm just using an example. If I have a protein A that is red 55, then I take a human protein and I find its homologs in other organisms. And in this case, maybe it's pretty straightforward because each organism just have one homolog. Then in the case of protein B, for example, if it's red 57, maybe it's not so straightforward because in each organism, you may have two homologs. Then you just need to do a little bit of careful work to classify the evolutionary relationship of protein B. So if you build a tree of protein B, you may see, okay, these proteins, they seem to be one like subfamily of this big family. And then the other subfamily is what we call paralog. So paralogs, they usually will, they originate mostly by gene duplication. And after duplication, it's likely they have adapted to different function and to interact with different partners. That's why for this kind of prediction, we tend to take only the orthologs, which are the equivalent proteins of what you are interested in. So once you do this, so it is quite easy for me to do this because the Grecian lab is actually mostly doing protein classification. So and for our genomic work, we also frequently do protein classification. So this is just all what I'm very used to. So after we did this, then we can connect proteins from the ortholog proteins from the same organism and convert this to protein problem perfectly to a standard problem of just one multiple sequence alignment and deducing the co-evolution. So, and we decided to go a little bit more ambitious. So instead of just taking like proteins that we sort of know they should interact and model their protein complex, we decided to maybe we can just do it on a proteome scale. So why did we decide to do this? So first of all, I thought like, for example, structure prediction is a very well-defined problem. And that's why AI is very good at solving it. But if we go to some that less well-defined problem, maybe humans still have some advantage. That's one consideration. So another consideration is there are a lot of large-scale experimental screens on protein-protein interactions. So they are very, very powerful. So they provide a lot of data for our to benchmark our study. But on the other hand, they also have their own problems. So I'm just listing two most popular large-scale experimental screen. So one is bacterial or yeast-2 hybrid, where you just fuse your two candidate proteins onto one is the DNA binding domain, and another is the activation domain of a transcription factor. So if your two proteins would interact, it will bring the DNA binding domain and the activation domain together. So then that can turn on the expression of reporter gene. So then that way you can detect the interaction. So another is just mass spec. So you let the cell and you do some cross-linking to make sure that things that are supposed to interact, they still maintain together. And then you analyze the mass spec, you get what proteins interact. So, but both of these experimental methods, they suffer from some significant false positive rate, because the proteins, they are sticky in non-physiological conditions. So for example, most proteins, you can crystallize them by forming some interchain contacts that are not really present in cell. So this false positive make different screen data set don't have a good agreement between each other, and it also made it harder to interpret and to trust the data obtained from these very valuable experiments. So then what I was thinking, okay, interactions identified from residue coevolution at least can compensate these experimental screens, because these coevolution, they are raised from some in vivo signal, like through a time of evolution. So obviously our screen also have our own false positives, but hopefully it's not likely the same kind of false positives as the experimental screen. So there are some challenges to the coevolution-based PPI screen. So the first challenge, it is computationally expensive, because we are studying one pair of protein at a time. And for E. coli, so which has 4,000 proteins, it will translate to 9 million protein pairs. And for eukaryotes, for example, human, it have 20,000 proteins. It will translate to 200 million pairs. So it is very expensive computationally. And another is that the signal to noise ratio is very low. So for example, E. coli has 10,000 real protein interactions, but it has like 9 million pairs. So that means the signal to noise ratio is about 1 to 1,000. So then the method to overcome the first limitation is we are not really trying to like screen every pair using the most accurate and not so fast method. So we basically designed this a hierarchical screening approach. So we will put those less accurate and faster method as our first stage, and we put the slower and more accurate method in later stage. So then every pair will go through the first stages, but only more confident pairs that has high hope to interact will go to later stages where we use slower method. But these different methods for different stages keep evolving as the field you want a newer method. So when we just set up, then we will start with some local statistical method. Question?
No question right now.
Okay. So we will start with some local statistical method and go to like global statistic and go to docking. But currently, due to the revolution of deep learning method, this completely changed. So all our step was updated to deep learning, various deep learning methods in our recent work. So, and another thing is important to monitor your accuracy throughout the procedure. So to do that, we need to have a set of proteins that we are very sure they interact, and a set of proteins we are very sure that they mostly don't interact. So this is the positive and negative control set. The positive control set, we took the consensus between different resources. So as I mentioned, because PPI data is quite noisy, then you cannot rely on single resource and believe they definitely interact. That's why we took a conservative approach and only took interactions that are approved by many different methods. And the negative controls, they are protein pairs that by any experimental evidence not yet known to interact. And we chose the negative control to be about 1,000 times larger than the positive control. So that we are mimicking what is happening in real case, where you do expect 1,000 times more more non-interactors than interactors. So, and then we use precision recall to measure the performance. So I don't want to go to very detail explaining what it is. And but for those that you are not very familiar with the precision recall, then I still want you to have a intuitive sense about how this curve should look like. So if you have a terrible predictor which doesn't have any distinguishing power, then you will have a curve that is very close to the x-axis. So that means terrible performance. If you have a perfect predictor, then you will have something that is pushed far away from both axes. So then as we go through different steps of our screen, we monitor the performance. So this is our old version of screen, this mutual information, and we are monitor the performance by the precision recall curve I just mentioned. So the baseline would be just random selector, then it's the horizontal line near axis. Then by using mutual information, especially some improvement over mutual information, we are getting a curve that is departing away from the axis, which means a better performance. And then as we go to global statistics, then with some smart treatment of the data, then we can get some constant improvement in our accuracy. And then in the final stage when we try to do docking, so basically the previous method you just identify the interacting residues. You don't know whether they will fit to a protein complex when you put it into 3D contact. So in the last stage, we try to put proteins into a 3D context and see if the co-evolving residue indeed can be like on the same protein-protein interface. That is also quite powerful and improved our performance. So obviously now all these things they are done, they are all integrated together by the deep learning network. Although so inside deep learning network, it still sees the co-evolution information, and it also sees whether the 3D structure of proteins can come together nicely. So and when we just started at 2019, maybe 2018, so there aren't many methods that are really doing PPI screening in this way. Basically, we are trying every pair and trying to basically do residue level co-evolution on every pair, and then extend to the full genome. So since there aren't other methods that are similar in nature, so we decided to, well, maybe we should directly compare to experimental screen, since that's how we decided to start. And so what we decided to do, we just took the benchmark set from those EAST hybrid screen and the mass spectrometry screen, and just compare our method against the experimental screen. And it is a little surprising to us that actually our co-evolution screen by itself is comparable or better than experimental screen. So here, so green, it means better performance, and red means worse performance. So on some data set, we are not doing the best, but in most data set, we are doing better than experimental screen. And if we combine co-evolution screen together with some experimental data, then we obviously will get the best performance. So, and then we will have this method, and then we just apply to every protein pair in E. coli that had a thick enough alignment. Then after that, we look into the pairs we get and see if we can gain any functional insights about the protein complex. And this is the example that I liked the most from that study, where we reviewed a self-inhibitory mechanism of DNA polymerase 5, which is a DNA polymerase used in DNA repair. So this DNA polymerase 5 consists of two proteins. So one is a catalytic protein, another is an inhibitory protein. So although the individual structure of those two proteins can be modeled, but how they interact wasn't known at that time. So then according to co-evolving residue, which are indicated by those red bars. So red bars are connecting co-evolving residue. And this dark blue sphere is basically the catalytic site. So you can see this interaction, if it happens, it will block the catalytic sites. And however, this inhibitory protein has another known conformation from the protein data bank, which is a dimer of this protein. So this dimerization happens upon activation by REC-A, by other protein. And upon activation, this yellow protein will form a dimer, and in this dimerized form, this strand that is blocking the active site will be kicked away. So that explains how this complex can become active. So then in our conclusion, we also predicted some novel protein-protein interactions. And here I'm just showing a couple of examples of those protein-protein interactions. And there are quite a few interesting ones that reveal some surprising link between the metabolic enzyme or transporters with some proteins that can control the either gene transcription or gene translation. And I think these crosstalk are unnecessary because so you need to have some link between the metabolism status to the regulation that control the transition from growth to stationary phase in bacteria. So, but these are not really experimentally validated, and but I'm just feeling very exciting to find possible new connections. But there are some of our predictions that we did validate with experiments through collaboration. So after setting up the initial pipeline, we decided, okay, we can apply this pipeline to a series of pathogenetic bacteria. So then we applied to basically all the most of the widely studied human pathogens, and then we identified some, also identified unexpected links. So for example, here I'm showing two examples, and these are also liked by some of our microbiologist collaborators. So they went on to test them experimentally and doing some follow-up studies. So, however, although our method was okay to in bacteria, but it wasn't. We couldn't use it to eukaryotes until recently. So why is that? Because our method still rely on the our initial method still relies on the coevolution, which will need 10,000 about like 10,000 proteins to diverse proteins to make a reasonable confident prediction. So you simply don't have that many sequences for eukaryotes. However, the deep learning methods greatly reduce the needed number of sequences. So now we can, with this method, we can move from prokaryotes to yeast. So again, I'm showing the precision recall curve. So, and this is some benchmark done on yeast. So in East, we upgraded our screen. So this in the first stage screen, we used a faster version of Rosetta Fold. So here you can see I show the performance of several methods. So this gray line, so which is DCA, this is a global statistical method, and you can see it is not very close to the axis, means it is not having much power. However, as we update to the deep learning tool, we are starting to get much better performance. And it was to our surprise that if we use our fast version of Rosetta Fold to do a first stage screen, and then using the much slower AlphaFold to do a second stage screen, we could reach almost the perfect precision, although the recall isn't so good. So recall is basically means what % of true interaction you can recover as in your prediction. The precision means basically indicates out of what you predict to be true interactors, what % is right. So this means although we cannot really recover a high rate of true interactions, whatever we predict to be interacting, it has a very high chance to be right. So, and with that, then we have a couple of applications. So the first is just to do a de novo screen to East. So then obviously out of whatever we can identify, there will be a very significant fraction that was already determined experimentally, having the experimental structure. Then there are also quite a lot that although the experimental structure wasn't known, but the fact that the interact was revealed at least by some large-scale experiments. And there is just a small fraction that will be known because East is such a very well-studied system. And another application that is very exciting to me is we can use our screen as a filter for the large-scale dirty experimental data. So here we basically tried to take candidate protein-protein interactions from different resources and try to use actually AlphaFold to evaluate whether AlphaFold also tend to support this interaction. So then as you see, if we take like gold standard, then AlphaFold would give support for about half of them. So these are definitely true strong interactions. Then you can have 50% of them predicted by AlphaFold. But as you go to either weaker or less confident interactions, the rate of interactions that will be supported by AlphaFold will reduce. So curated data set is just some from literature, mostly low throughput experimental studies. And you get more than 30%. And the Belgrade also took the very confident interactions supported by multiple literature, and you also get about 30%. But just in sets of large scale experimental screen, then you get only 20% that are supported by the AlphaFold. So then we also have different version of our fast version Rosetta Fold. If we make a more stringent version of fast Rosetta Fold, then we have 30% supported by AlphaFold, which is it's comparable then to low throughput experiments. If we lose our cutoff, then we can get a set that's comparable to a high throughput experiment. So, and another important factor is whether we can predict the 3D structures of the protein complex correctly. And due to the broad recognition of these deep learning methods, good performance, I guess it's not too surprising that mostly when you can detect the interaction, then it's easier to predict the 3D structures accurately. So we did our benchmark. So from this complicated plot, I just want you to zoom into one point. So what does this point mean? This means for close to 90% of protein complexes, our interfaces is at least 60% correct compared to the experimental structure. So here I'm just showing a couple of examples from the cases that we look at now, and to show that our prediction will closely mimic experimental structure. So here is a structure between ESR1 and ESR2, and this conformation closely mimic the ESR1 dimer. So, and also I'm showing an example of EGFR. So this is experimentally determined structure, and this is our prediction. So you can see, although so this structure has a lot of loop, and it's still quite accurate. The 3D structure is still quite accurate. So and one important thing is, by doing this large scale PPI screen and model the 3D structure, we can provide some structural insights into important protein complexes. So here are some complexes that we modeled in our yeast work. So the first is a signal peptidase complex. So this is our predicted complex. This is some picture I took from a bioarchival paper that is not yet published. So, and you can see our model really look very similar to the experimental structure. Then another is this complex involved in this intracellular trafficking. So here, this is our model, the protein complex. Well, so this protein complex has a lot of flexibility, but we are happy to see that we did recover this Y shape of protein complex that is observed in some low resolution images of this complex. So our most the case that we like the most from this study is this GPI transmedase. So this is a 5-component protein complex, and this is our model. And there was a recent study that published this structure. I don't know if it's published or not, but our collaborator did send us an experimental structure of a human version of this complex, and they look very similar to our prediction. But our prediction was sufficient to explain the catalytic mechanism. So for example, you can see here the catalytic residue is here, and near the catalytic residue there is a channel to allow the substrate to go through. So, and this is another interesting example, which is a protein complex between RAD55, RAD57, and RAD51. So RAD51 is this protein that is used in DNA in homology recombination-based DNA repair, and its function is to wrap around the single-stranded DNA in the process. So, but it is known that RAD55 and RAD57 will promote this conformation of this filament conformation of RAD51, but it's not known how exactly this process happened. So, but our structure did provide a mechanism for this. So because the interaction between RAD55 and RAD57 is compatible with the filament structure of RAD51. However, there's possibly another conformation of RAD51 that is at least observed in archaea, where the RAD51 just form a 6-mer circle, and apparently this complex wouldn't be compatible with this donut shape, but will be compatible with the filament shape, and we think that's why this interaction can stabilize the RAD51 in this filament shape and preventing it from going into the donut shape. So then what I'm doing now is to move from yeast to human. So it's actually not really, although our methods work quite well in yeast, it doesn't work that well in human for a couple of reasons. So first of all, humans, you don't have, so human has 4 times more proteins than yeast, and a lot of those proteins originate from more recent duplication. You cannot make as thick alignment as you can in yeast. And another thing is the human protein has a lot of those disordered regions, flexible linkers, and they tend to mess up the screen. We are still working on how to reduce this noise and make this method more reliable in human. but we did do some smaller scale application to human proteins that are potentially involved in cancer. So cancer drivers are frequently hubs of protein-protein interactions. So for example, we are just analyzing the large-scale protein-protein interaction data. Then for the 20,000 human protein, we totally can have like 700,000 putative PPIs from databases. And if you just look at the cancer driver proteins that at least currently considered cancer drivers, according to Cosmic database, then there are about 600 proteins, but they have like 100,000 putative PPIs. So they really tend to be hubs of PPIs. However, if you look at those experimental data of PPIs, then what you frequently see is really just like everything is cross-connected with each other. And I think this view isn't really correct because it's very much affected by those proteins, those sticky proteins that tend to form a hub of false positives. So they kind of glue every proteins together, even if they are working on completely different pathway, then they are all clustered together. If you look at the data with a very high noise. So what we did is similar to what we did in East, and we just applied our pipeline to these human these cancer related PPIs. Then we are just showing that indeed applying this computational method can help you to distinguish true interactions from random protein pairs, although the distinguishing power is much worse than what we see in East without due to all sorts of problems that I mentioned before. And not surprisingly, so when we do see a good signal by our deep learning methods, then we can model the 3D structure of the protein complex rather accurately. So then here is just our modeling procedure. So we basically get the sort out the orthologs, and we generate paired multiple sequence alignments. So this is our traditional coevolution method. And on the other hand, we are mining all the PPI databases to get the interactions, the candidate interactions that involving any cancer drivers. And then we skip the Rosetta fold part because this is a relatively small number, and we can model it all with AlphaFold. And then we model the a significant fraction with AlphaFold, and then if we get a confident protein complex, then we study it manually. So then after sort of cleaning up this PPI data with this deep learning method, we are able to obtain in my eye, a more reasonable cancer interactome data. So basically, we clustered the proteins using the supported interactions by our method. Then you can see they do cluster more closely by function. And all these functions, they are not surprising. They are all very well-known processes that are involved in cancer. But I just like the fact that they did partition into functional group. So, and then what we did next is to integrate our cancer interactome data with somatic mutations that are observed in cancer. So, and then what we did is basically to take all the somatic mutations observed from all those large-scale sequencing of cancer patients and map them onto the protein complexes. And then we can analyze if there's any complex that are enriched in somatic mutations in cancer, or there are any interfaces that are depleted of such somatic mutations. So then we indeed see such complexes, and then we do see that complexes that seem to be enriched in interface mutations, they show a different functional group compared to the complexes that tend to have a depletion of mutations in the interface. So those that so these are the functions that tend to show enrichment in mutation. So they are involved in cell division, signal transduction, or cell differentiation, and some cell defense. So these are, if you think deeper about these functions, they are basically a function that cancer cell either doesn't need or they need to rewire it to facilitate their unlimited growth. And on the right, I have some processes that are still try to avoid the mutations. And these are the essential functions that cancer cells seem to be also need. For example, transcription, DNA repair, especially DNA repair, because for cancer cell to survive, they need to survive those radiotherapy and chemotherapy, which mostly just to cause DNA repair. If the cancer cell doesn't maintain a good DNA repair, cause DNA damage. I mean, this treatment cause DNA damage. That's why DNA repair may be even more important for cancer cells. So here I'm just showing a couple of examples of interfaces that are either enriched or depleted of somatic mutations in cancer. So on the left is interfaces that are enriched, and those red balls are basically mutation hotspot. So sometimes this enrichment is just caused by one mutation hotspot that is quite well known even without our structure data. However, some interfaces, they don't seem to have a mutation hotspot, but when you model the 3D structure, then you can see maybe there are many residues across the same interface, they are mutated. Then that also suggests that that interface may have a role in cancer progression. And here just some DNA repair proteins. So I think, well, not all of them, but at least this is a DNA repair protein, and some other proteins that tend to not have mutations in the interface, meaning maintaining this complex is still important for cancer cells. So here is just another example showing that even for known protein complex, so this is a known protein complex. However, there is a part of the interacting motif that is missing from the complex, which we can recover from our analysis. Just extending the known interface to some other parts of the protein. So here is just if you look at those proteins in terms of protein network. So this is a network that is centered around AKT, which we took from KEGG. Then we were able to recover its interaction with several important partners as circled here. And this interaction between AKT and its partner is happening through a short sequencing motif in the other partners. So although this motif is mostly disordered and it's quite short, but the coevolution, the deep learning method still can recover this interaction. And this sort of a big protein and short motif interaction is observed a lot in human, and quite a number of them can be detected by the deep learning methods. Even if sometimes that motif has a phosphorylation site, and AlphaFold doesn't really know it has a phosphorylation site. But I think using some coevolution signal around the phosphorylation site, it still can recover that interaction. So yes, so this is another example of that ESR1. It's also interaction hub interacting with many other proteins that has a short LXXLL motif, and we can see that using AlphaFold analysis of our sequence alignments. So with that, I'd like to thank my lab members. We still have a small lab. So Jing and Jimin. So Jing is my postdoc. Jimin is my research scientist. They did most of those work I mentioned are done by them. And Jessie is my first grad student, and Aaron is our system administrator. So we still work very closely with both my grad school advisor and postdoc advisor. So grad school advisor, we still work on genomics, evolutionary genomics and comparative genomics. And with David. So we work on like using the tools that are developed in his lab to for various application in the biological systems. And I also like to thank many of my collaborators, and our funding is mostly coming from my startup package and some training grant to my people.
Thank you, Qian. Thank you for this very nice and clear presentation.
Thanks.
So I think we'll take first, I don't see any question in the chat, so we'll take questions directly. So if you have a question, please raise your hand. Shoshana?
Yeah, you can skip. I have many questions, but I'll ask just one to begin with. How exactly did you use AlphaFold 2 in your pipeline? Did you use it in the use it to predict the actual interaction or the independent structure? Or did you actually also use the AlphaFold multimer version that was trained on complexes? And how did that perform if you did use it?
So in our East work that is done before AlphaFold multimer. So for our, so at that time we didn't use, but now we are always using both. So usually, so what we see is like, so when the two proteins have a large and bona fide interface, then both AlphaFold. So then probably AlphaFold multimer will do better. However, if you have a lot of those loops in there that's supposedly not to interact, then AlphaFold multimer may still force it to interact. Then your protein just doesn't look like right. So the interface is still there, but it's like a exaggerated interface. It will look larger than realistic. So basically, it seems like both AlphaFold and AlphaFold multimer have their advantage. We usually use it in parallel, and if we see a consensus between the two, we are more confident.
And how did you interleave it with Rosetta Fold? This was not very clear.
Oh, so Rosetta Fold is constantly developing. So in its initial form, it is still much worse than AlphaFold in predicting the 3D structure. But the very recent version is comparable to AlphaFold. That's why in the initial version, we still rely more on AlphaFold to model the 3D structure. But the use of Rosetta Fold is just Rosetta Fold is our own tool. Then we know how to twist it in the best way. So we want something that is very fast. Then we modify Rosetta Fold. We know, okay, this is important, that is not important. Yeah.
Okay, thank you.
I have a question, Qian. So you mentioned that there are several, that you recover 30% of the real protein-protein interactions. Do you know if whatever you can't recover followed because AlphaFold cannot detect them, or it's very followed earlier in your pipeline?
That's a very smart question. So yeah, so let me show the curve. I think it will explain. So exactly because we are thinking, well, maybe we made this very fast version of Rosetta Fold, then we are missing most of them just because Rosetta Fold missed them. So that's why we tried to apply all the gold standard to AlphaFold. And you can see AlphaFold will still miss 50%. And not to say this gold standard is really strong interactions. Most of the transit interaction wouldn't even go to gold standard. That means true transit interactions, AlphaFold probably will miss like 80 or 90%. So strong interaction will miss a lot. So, and not to say this is the data in yeast, if we do it in human. So in yeast, we can recover maybe 50%. In human, we probably can only recover 10%. So we are still 10% of good interaction. So if you consider weaker interaction, then our recall will be like about 5%. That's why we are still struggling with this study on human. Just you know there are many more interactions, but due to all sorts of limitation, we don't yet see it.
And is it possible to structurally analyze those interactions to, I mean, is there anything that can, between the predicted and non-predicted interactions in terms of structure and structural property that can explain why they're missed by AlphaFold?
That's a direction that we are very interested in looking into, but currently I don't really have a lot of clues. So I have one example where, so for example, so this is from a collaboration. So where like human protein, they have like 3 versions of a similar proteins, like gene expansion. Then what I see is if I use each of these 3 versions, just taking ortholog of that small group, I'm not going to get the interaction. However, if I just consider, well, all 3, they come from the same ancestor, then I just combine all of them, I was able to recover the interaction. So at least one fact is, like if you have very shallow sequence alignment, then you are not going to get it. But the problem is, of course, if I don't distinguish that 3 versions of the same protein, then I'm trying to assume they all interact in the same way. If my assumption is right, then this is the right way. If my assumption is wrong, then it is not. So we don't really know like why things that except this, we don't really know why things that should interact don't show a signal, but we do know better about why things that shouldn't interact may show a signal. So frequently those like hydrophobic and disordered regions, they may somehow still have some sticky effect, especially in AlphaFold that's trained to recognize hydrophobic interfaces.
And then, Shoshana.
I mean, it will be good if other people also ask questions, but I just, it would be interesting to know the predicted structures from your screens. Do you make them available? Do you make the data available publicly?
So for East, we deposited in this model archive, which is something developed, partly with PDB.
The PDB in PDB-KB, is that it?
No, it's called model archive.
Model archive, okay, yeah.
So I think that's the place we probably eventually will deposit all our models to.
Also is the PDB-EKB, they have the Beacon project, which is very similar. So it would be nice to find out.
Yeah, I think that model archive is somewhat also related. I think they
related, okay. Yeah. Okay, yeah. My other question is also quite general, in terms of computational, resources, how heavily dependent is your approach on these resources, and what resources do you have access to that allow you to do this?
So the thing is, so if you know exactly what to do, then the resource then you don't need that much resources. But in this experiment trial and failure process, you do need a lot of resources. So that's why a lot of these things, they are still done in collaboration with the Baker Lab, which has much more resources. And what we use is at least for the East project, we use the Azura, so Microsoft's resource, which they had some donation to the Baker Lab. So it's like, yeah, but it doesn't need that much resource, but I guess still you will need to have like on the order of 50 to 100 GPUs to do it in a reasonable, like timely fashion.
Yeah, thank you. This question is important in view of what you would like to do in the future.
Yeah. But I think, yeah, so basically those like cloud services, they are quite good, but my lab also try to use it. But the problem is like they usually they will charge you, for example, if you forgot to delete your data, and then every day your data is there, it will charge quite a lot. Or you forgot to log out, so it is a little hard without some.
You have to have a good IT admin that helps you,
Yes, help to make sure that.
Lots of questions, but any other questions, participants, please come forward. It's an opportunity to ask any question. Doesn't really matter. Okay.
I have a question, Qian. So, Qian, it was my understanding that if you put two proteins, if you use two sequences as input to AlphaFold, it will still try to sometimes force an interface even when there is none. And I think that was a question to one of our previous speakers from this audience is, how do you, is there any metric that you can easily use to differentiate a real interface between from a false positive based on AlphaFold prediction?
So yeah, if you just look at the PDB output of AlphaFold, then even if AlphaFold isn't so confident about the interaction, it may still just show an interface. So that's why it is more helpful to look at, beside the PDB, AlphaFold will have some like histogram, or some, you can also output that paired alignment error file. So they do have their additional like estimate of the probability, and we do use those files to evaluate whether it is an interface. Yeah, but even if you have high confidence by those files, there's still quite a lot of false positives. Yeah.
Maybe I just can add to this, because we recently looked for the correlation between the IPDDT, that was produced by AlphaFold on their, reliability score for the interface with actual other physics-based scores that people derived in Capri. And there was a poor correlation. The correlation was much poorer than between other scores. So there is still a disconnect, between these reliability scores and physics scores. So I think this could be used in a way to find out, these types of poor correlations.
Yes. So we use that. So PRDT is like a residue.
Yeah.
Per residue score. So we use most of those residue pair scores.
Right. That's what that they use that as well.
Oh, I see. So it's also not very good.
No.
It's good to know.
Okay, that was a fantastic overview. Thank you. This work is really, really exciting, and hopefully this will progress in the future. But the data, the experimental data is very, very noisy as well.
Yes,
and,
that's why I say it's a poorly defined problem. If you want to train a network, then you don't know what to trust.
Yeah. So that's going to be a big help. Okay, I think, Anastasia, what do you think? Shall we?
Yes.
Someone is asking a question. Oh, no, doesn't ask a question.
Thank you very much, Qian. That was really fantastic. Thanks.
Thanks.
Wonderful, and we are happy, we caught you at the end.
Oh, thanks. It was
terrible to meet you. So, good luck to you.
Thanks, everybody.
And thanks for the
Hope
time.
to see you.
Thank you, Qian.
Anastasia, are you coming to the Rosetta Con?
I think so, yes.
Okay.
So I'll see you there then.
Take care, and thank you again. Thanks to everybody that participated, and hopefully next time you can ask more questions.
Don't be shy.
Don't be shy, yes. Thanks.
Thank you.